lysosomes or the cytoplasmic cell organelles. These cell organelles generally arise from either endoplasmic reticulum or from Golgi complex. And these lysosomes are the part of endomembrane system, because as like the Golgi complex and endoplasmic reticulum, these are the part of endomembrane system, because the functions of these cell organelles are in close coordination with other cell organs like endoplasmic reticulum and Golgi complex. That's why it is a part of endomembrane systems. And here generally it is spherical or round shaped structure, but sometimes, rarely, like in the root hairs of plant cells, so it is irregular in its shape. Root tip cells, the type of variation can be seen in the shape. And here these are structures which are made up of single lipoproteinaceous membranous structures. That means these are surrounded by single membrane of lipoproteins, and ranging from 0.2 to 0.8 micrometers in their diameter. So these lysosomes are arising from the maturation side of Golgi complex, and these lysosomes were first actually discovered by Christian D. Dewey scientist. And these are their number will be ranging, or actually the number will depends upon the metabolism or the functional activities of this particular cell. Generally the one which are metabolically active, those cells contain highest number of lysosomes in them, particularly like the cell which is undergoing division, or the meristematic cells which are repeatedly undergoing division. So repeated mitotic division or is a type of highly metabolic active mechanism. That's why in such type of cells the number of lysosomes will be very high. Then these lysosomes are very specific in their nature, because they are having various types of enzymes present in them. Nearly 50 types of enzymes are present, but it is not compulsory that all 50 types should be present in each and every lysosome. But the overall, if you see the number of enzymes, the 50 types of enzymes are present in the lysosomes. Let's take some of the enzymes which are present in the lysosomes. Phosphatases, nucleases, carbohydrases, lipases, sulfatases. Many such type of enzymes are present inside the lysosomes, which are concerned with various metabolic activities. Like nucleases are there. So these nucleases will act on the nucleic acids like DNA. So, and carbohydrases which are acting on the carbohydrates, lipases which will act on the lipids, and proteases which will act on the proteins. Likewise, various types of enzymes are present in the inside the lysosome, which is concerned with various types of metabolic activities. So lysosomes are the structures which contain thin membrane structure, and it will not release the enzymes which are present in it into the cytoplasmic content, but whichever the structure have to be digested, that structure will enter into the lysosome. There the hydrolytic enzymes which are present inside the lysosomes will act on them and digest that particular material, and whatever the undigested material which is left out, which will be released outside by exocytosis method. The enzymes which are present in the lysosomes are active in the acidic condition. So here acidic conditions are maintained inside the lysosomes by accumulation or influx of the H+ ions into the lysosome, so that the acidic conditions are maintained inside the lysosome. So here as we said that lysosomes are formed by the cumulative effect of both the endoplasmic reticulum and the Golgi complex, and here the precursors of hydrolytic enzymes. The precursors of hydrolytic enzymes are formed in the endoplasmic reticulum, where they are organized into a spherical structure with a formation of a membrane, so that they are converted into the lysosomal structures. So here lysosomes or the hydrolytic enzymes are very active. the activity of the hydrolytic enzymes are restricted by formation of a membrane around them, so that the other cellular contents or cytoplasmic contents which are present are protected from the hydrolytic enzymes. But when any cytoplasmic content have to be digested, so those structures will enter into lysosomes, and there inside the lysosome they are acted upon by the hydrolytic enzymes. But at sometimes what happens is the membrane present around the lysosome will get burst or get delicated, so that the hydrolytic enzymes are released into the cytoplasmic content. Once the hydrolytic enzymes are released into the cytoplasmic content, so all the cellular components have got digested, that leads to the death of the cell also. So in order to protect from such unfortunate condition, the hydrolytic enzymes are restricted by formation of a membrane around them to form into a lysosome. So here lysosomes are generally polymorphic cell organelles. So polymorphic means the structure which exists in more than or many morphological organizations or morphological structure. Based on the morphology and function, actually 3 types are there, but additional to the 3rd type, the 4th type is also there. Let's discuss the 3 types here first. So first one is called as primary lysosomes. So these are the lysosomes which are formed from the endoplasmic reticulum or the Golgi complex by forming a membranous structure. Generally these are spherical shaped structures which are having single lipoproteinaceous membrane along with the hydrolytic enzymes inside them. So these are formed towards the Golgi vacuolar maturation side, or we can say that some of the Golgi vacuoles are converted into lysosomes. Those lysosomes are called as a primary lysosomes. So here the hydrolytic enzymes which are present inside the primary lysosomes are very active. As and when any food material or any foreign particle enters into the lysosome, there the activity of the hydrolytic enzymes will start. So then they are converted into second structure called as a secondary lysosomes. So these second lysosomes are also spherical shaped structures which are having lipoproteinaceous membrane along with the hydrolytic enzymes, as we have seen in the primary lysosomes. So it contains hydrolytic enzymes along with the food particles or the food materials or the foreign particle or any substance that have to be digested will enters into the lysosomes. Now that structure is called as a secondary lysosomes. Now the digestion of that particular material will start inside the secondary lysosome, or the activity of the hydrolytic enzymes will start in the secondary lysosome, where the food particle will gradually start digested. Then after digestion, now the digester material will be released outside, that will be used for the new cell components or the new cells. Then the remnant which is present is converted into a structure called as tertiary lysosomes. This is also nearly spherical in structure, which contains lipoproteinaceous membrane, and they also contains the hydrolytic enzymes, but along with that one they contain undigested material. After digestion, when the digestion is taking place in the second lysosome, the digested material is coming out, and the undigested material will remain inside that lysosome. Now that lysosome we call it as a tertiary lysosome, or this is a one which is otherwise called as residual body also. Residual body. And here how does this undigested material have to be released outside means, this tertiary lysosome gradually move towards the cytoplasmic membrane or the plasma membrane, and it will dump this material outside by a process called as exocytosis. So let us take the diagram of endoplasmic reticulum. This is a rough endoplasmic reticulum part. and smooth endoplasmic reticulum. From the endoplasmic reticulum, we can see the formation of the Golgi apparatus structures, and the Golgi apparatus which contains the branched structures and also the unbranched structures, that is tubules and also the vesicles. So along with that one, we can also see the formation of the vesicles and also some of the spherical shaped structures which are separated out. So these structures are acting, some of them are acting as a lysosomes. Then these lysosomes which are spherical in their structure, which contains hydrolytic enzymes, now we call them as a primary lysosomes. So if any food particle or if any foreign particle, so this will enter into the primary lysosome, then it will be converted into secondary lysosome which contains the food particle. Now we call this as secondary lysosome, and this secondary, inside the secondary lysosome, as we had discussed, so the digestion of the food particle will start. That is a activity of the hydrolytic enzymes will start inside this one, and the materials, digested materials will be released outside, whereas the undigested structure which is acting as a tertiary lysosome which contains the undigested metals will be moving towards the plasma membrane, where consider this as the plasma membrane. So here it will be coming towards the plasma membrane, where it will be dumping out all these materials outside to the cell. This process is called as exocytosis. So this is one process where the different structures are of the primary lysosome are coming here. And one more method is there. How does the foreign particles will enter into the cell? So it occurs either by pinocytosis or phagocytosis. Pinocytosis means any liquid material, or we can call it as a cell drinking. Any liquid particle is entering into the cell. Consider any liquid material is coming into the cell. Pinocytosis. Or we are showing any solid particle which is coming into the cell. So we call this as a phagocytosis. And when because of pinocytosis, a vacuole like structure will be formed. So we call this as a pinosome. And this solid material containing one is called as phagosome. Then the pinosome or the phagosome, so it will be converted into the lysosomal structure, where the activity of this hydrolytic enzymes will be acting on it. And once again from here, the digested material will be released outside, and the undigested material, that is what we call it as a tertiary lysosome, will be formed. So here this tertiary lysosome will be releasing the undigested materials towards the outside by exocytosis. Likewise, the lysosomes are not only concerned with the digestion of food particles, but also concerned with the digestion of various liquid contents which are entering into the cell, and also the solid metals which are entering into the cell. That is simply to say, they are concerned with digestion of various types of materials, either it may be a food or any foreign particle or any harmful substance or any liquid content or a solid metal which is entering into a cell is digested by the hydrolytic enzymes, and those the digestion is taking place inside that lysosome. So this is a intracellular digestion which we call it as. So likewise, the three types of lysosomes are formed in the due course of the changes which are taking place from primary to secondary, secondary to the tertiary. Secondary lysosomes are otherwise called as heterophagosomes, because they contain different types of materials which are coming into that one. So we call such secondary lysosomes as a heterophagosomes. Then others are also there, those are called as autophasic vacuoles. Or we can also call as autophagosomes. So what is this autophasic vacuole or autophagosomes means? Actually, as I said, that whenever a particle or any substance have to be digested, it will enter into the lysosome. But actually, but sometimes what happens, the hydrolytic enzymes which are present inside the cell will be releasing outside into the cytoplasmic content, which will kill or which will digest the injured cells or the cells which had become aged. So such type of cells have to be digested. So that is done by releasing the hydrolytic enzymes into that injured or the aged cells. So that is what the digestion is taking place outside that lysosome. So this type of vacuoles are called as a autophasic vacuoles or the autophagosomes. And in this process, the injured cells are completely digested. That is, killing of the cell is taking place. So this is one condition. And other one is when the cell is in starvation. Starvation means whenever the cell is not getting the required amount of food material, what happens? It start eating the cellular contents or cytoplasmic contents. So the eating of the cytoplasmic contents will lead to the destruction of the functions of the cell, gradually leading to killing of the cell in which the lysosome is present. So that is the reason why these lysosomes are called as suicidal bags, which lead to death of the cell by itself. So that is one other character related to the autophasic vacuoles. So that's the reason why it is also considered as one form of structure exhibited by the lysosomes, apart from the primary, secondary and the tertiary lysosomes. So what are the functions concerned with lysosomes? So these are concerned with intracellular digestion, that is digestion of various metals inside the cell, and extracellular digestion, that is digestion of the some other particles outside the cell. Apart from that one, they are concerned with the digestion of proteins, lipids, nucleases which are concerned with the digestion of nucleic acids, peptidases, proteases like sulfatases, phosphatases. So all these are the enzymes which are concerned with the digestion of the respective chemical substance. All these are present inside the lysosomes. So those are the part of hydrolytic enzymes.